Start recording. Okay, good morning to those of you who've joined us for the Centers of Excellence in Hematology Monday Microscope, Microscopy Monday, as we call it. We want to welcome you and encourage you to share this opportunity with other friends. The Centers of Excellence in Hematology support around the country, and those cores provide microscopy, some of the cores. And this seminar series is to kind of inform you on those microscopy cores that are available, also give you the opportunity to, so to speak, in person, ask those microscopy core directors questions about the kind of microscopy tools there are and what you should be doing as good practice for your microscopy. Before we start this seminar, I want to share my screen and show you the website for the Centers of Excellence in Hematology. This is our website, which is CCEH.io. And what you can find here are a lot of useful tools as an investigator. First of all, the front page will show you some pinned announcements. For example, if you hit this top, Type A applications are open, it'll take you to a funding page. And I know this is going to launch. And what this is funding opportunities for you to obtain research monies to do a P&F project. There are Type A and Type B grants. Currently, the Type B applications are closed, but there will be another call in February. But Type A grants are available to apply for all the time. And what they are is essentially $5,000 to $12,000 worth of core credits. What you do is you apply, it's a one-page application, and you describe a core that you would utilize, and the money gets banked at that core for you to utilize. So for example, if you were going to do microscopy at Indiana with Ken, you might write a proposal that would you use the CODEX core that they have there, and you can apply for monies that would help provide the funding for you to get that very key preliminary data that helps you apply for those bigger grants. Let me get you back home, and then I want to show you one other thing. If you go up on the top of the website, there's a place that's called Services, and if you click that link, you'll find all the different cores that are supported by the Centers of Excellence in Hematology. So for example, how do I evaluate or analyze cells in a pathway? If you click that link, you can come down here and go cell analysis. And what you will find is the University of Utah has a metabolomics core, an iron and heme core. Indiana has a flow and tissue cytometry core, and their intervital and 2,3D microscopy. Also single cell proteomics. There's flow cores at Cincinnati and Indiana and Yale. There's morphologic analysis. And I just encourage you to go to our website to find out more information, because really these centers are for you guys, for researchers, to find those cores that you're not able to have at your particular institution, but are available across the country to investigators to give you state-of-the-art tools for developing your projects. And I'm going to stop sharing there. Actually, I'll keep sharing for one more minute. So the Microscopy Monday seminar series is the second tab down on the homepage. And if you click that link, you'll find Ken, who's going to present to us today. And he comes from Indiana University School of Medicine. He's a professor in medicine there in biochemistry and molecular biology. And today's talk is going to be CODEX Multiplexed Tissue Cytometry. And Ken, we want to really thank you for presenting today, and we'll let you share your screen and take it over. Just so that individuals know, you are welcome to populate questions in the chat. You are also welcome to interrupt and ask questions. Any of that is just fine. And this is really an informal sort of opportunity for you to really get educated in a safe space about good microscopy skills.
Safe space. Can you see my my slide?
I can see your slide, and you're laughing because it's not a safe space.
I don't know. I guess it's a safe space.
We're not hostile.
we'll see. Well, we'll see how it turns out. So
Okay.
anyway, thank you. And as I was saying earlier, there aren't a lot of us. So to the degree anybody in the audience has a question, they can pipe up at any point and interrupt me. I would love that, actually, because there's nothing I hate more than talking to computer monitors. So yeah, please, if you have any questions, let me know, right? So with that, great, it actually works. So I'm in charge of the flow and tissue cytometry core, whose goals are to provide flow cytometry and cutting edge microscopy services. And by cutting edge services, everybody probably has access to things like fluorescence, confocal, these kinds of things. And so there are two unique imaging services that we provide that you may or may not actually have access to at your institution. One of them is intravital microscopy, and I talked about that before. And the other one is this multiplexed tissue cytometry, and that's what I'm going to be talking about today. So one of the first questions you can ask is, why would you want to do tissue cytometry? What is tissue cytometry? Tissue cytometry is just like flow cytometry, except you have spatial information. And the idea of it is that it's all very well to understand what the cellular constitution of an organ or a fluid is, but you'd also like to know something about the relationship between the cells. So for example, these are some immune cells that are crowded around glomeruli. In principle, they're doing some immune surveillance around the glomeruli in a kidney. You wouldn't know that if you grind this up and just look at the individual cells. And the beauty of it is that if you start doing these studies at scale, and so with the development of techniques doing tissue clearing, we can image entire organs, entire mouse organs. And instead of just kind of like getting, I mean, I was raised on the representative field. You know, you collect a field of an image and you call that a representative field. And you can now census every cell in a tissue. This is some work that was done by Michael Gerner at Washington, and this is a lymph node. And you can see the complete analysis over here on the right, where he's got all these different segmented. You see these FlowJo diagrams, which are equivalent to what you do with flow cytometry, except they have the additional information of where these cells are located. So you can see, you can again, you're getting a complete census of the cells. You can detect and characterize rare events in a tissue that you might miss. You can characterize interactions and structures at multiple scales, the relationship between cells and vessels, glomeruli, whatever. And then finally, you can apply advanced methods of data analysis to identify unforeseen patterns. And that's going to be kind of a theme of what I'm talking about today, is that volumes of this size, you may very well go in with a hypothesis in mind, what you're looking for, but I can guarantee you're going to see more stuff in it. And when you start looking at these things with multiplexing, it becomes that much more rich and beyond what you're expecting to look for. So why would you want to do?
Ken?
Yeah.
I'm so sorry. I have a question.
Sure.
Is there a good clearing mechanism that doesn't cause autofluorescence in tissue?
That doesn't cause autofluorescence. I haven't really encountered clearing techniques that invoke a lot of autofluorescence, but I can talk with you about that on offline, I think, if that's okay. Yeah, no, there are plenty of them. I mean, it's daunting to look through them all. I can point you to a really nice review article on kind of looks through them. They have various strengths and weaknesses. So why would you want to do multiplex tissue cytometry? Well, by multiplex, what I mean is looking at more than 1 or 2 or 3 or 4 proteins. And obviously you can be greedy. You just want to characterize more cells and structures. But I think in hematopoiesis and in the field of the CCEH, you require multiple probes to identify cell types. I mean, this is some data that came from Daniel Lucas's lab, where he's using 20 or 30 different antibodies in a gating strategy to identify individual cells by their presence and absence of multiple markers. So at the end of the day, particularly for hematology, I think it's really crucial that multiplexing is used. So how does one do multiplex tissue cytometry? Well, most of you, probably everybody, there's 9 of us, are familiar with this, confocal microscopy. And this is an example of using, 4 different laser lines and 4 different emission filters to distinguish 4 different probes. That's pretty good. That's easy. And there is not much overlap in the excitation between these probes. There's not much overlap in the emission between these probes. So you can resolve them pretty nicely. But once you start getting into however many probes this is, the excitation and the emission spectra start to overlap with one another. So when you start exciting one probe, you're exciting more than other probes. You start seeing overlap in emissions between channels. And so it gets harder to distinguish these things from one another. And so a technique that's used to get around that is something called linear unmixing, where you have a sensor with a portion of a fluorophore fluorescence that shows up in other channels. And you can use one of these unmixing matrices to unravel that and to correct for that overlap. And this is an example of some work that we're doing at IU for the NIH Kidney Precision Medicine Project, where we're looking at human biopsies and characterizing, in this case, 7 different probes, as well as autofluorescence and second harmonic signals to characterize human kidney disease. And we use linear mixing of probes to distinguish these different fluorophores. And one thing I want to point out is that to do linear mixing, you have to have really, really clean data. And because the reason for that is that if you look at this graph here, this is Herxed in the blue channel and the fluorescence of Herx showing up in the green channel. And this is a bad example, but my point is this. If I want to say that, if I want to correct for the Herx fluorescence showing up in the green channel, I would take this 2500 and I would go over here and say, okay, good. I'm going to pull 800 gray levels out of my green signal and say that's crosstalk. When in fact, it's this much. And in the end of the day, you're overcorrecting some pixels and you're undercorrecting other ones. And so to the degree there's any noise in your image, you end up getting really kind of crappy data for a single pair of probes. So to the degree that you start putting that into these matrices, it gets really dirty really quick. And so it's something that it's a fraught technique. Another way of doing this, and I'm definitely not talking about this today, is to avoid the whole problem of fluorescence crosstalk and use imaging mass cytometry. And this is a technique where you label antibodies with heavy metals, and use and you basically go through with a laser beam or a MIDI beam, and you vaporize the tissue and you send it at the stream to a tissue cytop mass cytometry, and you identify individual heavy metals, and it's got micron level resolution. I think they've got 40 or 50 different heavy metals that they can use to distinguish these things. So it's very, very, very nice technique. It's an expensive technique, both in terms of the instrumentation and the reagents. Now, increasingly, people are using sequential imaging. And by that, what I mean is they're collecting, if there's a certain amount of overlap, they will collect 3 colors and then somehow eliminate those 3 signals and put another set on and then do it again and again and again. And one of the earliest ways of doing that involved labeling your tissue with 3 antibodies that would be directly conjugated to fluorescence. And then you would strip them off, various techniques where you can strip those antibodies off. This is a combination of glycine and urea to strip the antibodies off. You reprobe your tissue and you then do another 3 and you strip them off and you reprobe your tissue and do it again and again. This is not a technique that's really found much use, actually. Alternatively, and this is something, there's a commercial version of this technique where you label with sequentially, but now you're going to bleach the fluorescence out. So you label with 3 antibodies, each directly conjugated to a fluorophore. You then bleach the tissue, either chemically, which is what Ron Germain came up with using a really nasty agent, or you use light. And there's a commercial system called the Canopy system that uses photo bleaching. So you bleach out the tissue, throw another 3 antibodies on, image those, and you're off and you're doing that. It's another technique, and again, it's commercially available. What I'm going to talk about today is something called co-detection by indexing, or CODEX. And this is something that was developed at Stanford by Gary Nolan's laboratory. And the basis for it is this. If this is your antibody, I don't know if can you see my mouse?
Yes,
Good.
I can.
Thank you. So if this is your antibody construct, you're going to reduce the antibody and then do a malamide reaction to get a malamide modified oligonucleotide attached to it. This is a very specific sequence. You conjugate that. Now, at that point, you've got an oligonucleotide conjugated antibody. You take those oligonucleotide conjugated antibodies, throw them on your tissue, and what you're going to do is there are a set of fluorophores which are cognate for that particular sequence. They're going to recognize that. They're going to bind to it. You're going to add those. You're going to image the tissue, and then you're going to rinse those cognate reporters off. And so it's this reversible binding that it's very easily done, very easily accomplished. And then you strip them off and you do it again. This is a better description of how it exactly would work. You literally incubate your tissues with 20, 30, I'm going to show you an example of 50 antibody cDNA oligoconjugates all at once. They all go on the tissue at once. You add DAPI and 3 cognate fluorescent oligonucleotide probes that are going to specifically find those antibodies in the tissue, label them, and then you're going to collect a 4-channel image. You collect an image of DAPI, which is going to show you where your nuclei are, and it's used as a registration between cycles. And you collect 3 fluorescence images of the 3 cognate fluorescence reporters. You rinse, you collect that image, you rinse it, and then you strip off the tissue, the reporters, and you add another set of reporters on, and you do it again and again and again until you've run out of reporters. And that way you build up an image, if you will, from each sequence. So the sequence 1, you added 3 reporters here, you rinsed them off. Sequence 2, you added these 3 reporters, rinse them off, and you do that over and over and over again. And each time you use the DAPI labeled image to identify and register your sequential images. So this is an example of a field of cells. These are just in the nuclei. It's after addition of 1 probe, 2 probes, 3 probes, 4 probes, 5 probes. And this is something I'm always embarrassed by. I put this together a long time ago. I don't know what the tissue is. I know no idea where I got it from, but it gives you the idea of how you're building up an image over time after these events. Again, this was developed by Gary Nolan's lab at Stanford, and he's done some heroic work with it. This is an example. He developed a 56-panel antibody for analysis of colorectal cancer. So this is the same field image with 50 different probes using this CODEX technique. He conducted these analyses on 140 samples from 35 advanced stage CRC patients. And from that, he identified 9 conserved distinct cellular neighborhoods. And I'll explain more about what a cellular neighborhood is. But again, it's a spatially determined relationship between specific cell types. And he found out that when looking at these patients, which were not subclassified in any particular way, he found there was a specific cellular neighborhood correlated with survival in a high-risk data set, patient subset, which is really good news for that patient subset, which otherwise were completely undistinguished. And then he also found that there were some inferior outcomes associated with different kinds of neighborhoods. So in other words, he took a group of patients, a cohort of patients for which maybe there was no way of staging who was where and how severe their injury was, their disease was. And using this technique, he could distinguish them, if you will. So we've incorporated this technique at IU. We acquired a system, I don't know, 2.5 years ago, something like that, from Akoya. And we've been doing studies of kidney, colorectal tumors, pancreas, and diabetes. And these are examples of some images of spleen. And this is an example of work that we've done for the NIDDK for the Kidney Precision Medicine Project. And it kind of shows you how the sequential images can be built up to put together a multi-parameter image. And this is 23 different targets in kidney tissue. Now, kidney is really nice in the sense, visually, it's really nice because you've got these structures and it all stands out. It looks really, really cool as compared with, for example, other tissues where you've just got these individual cells that aren't forming specific structures. But it gives you a sense of how you build up an image this way. If you have any questions, I saw something in the chat.
That was just me telling people they could put things into the chat.
All right, good.
I have a question.
Sure.
Which is, a lot of individuals are doing single cell RNA-seq, so to speak, and sort of getting an idea of a lymph nodes transcripts that are present. How does that correlate at all with this protein detection piece?
Well, there's 3. I'm going to stop this because I'm going to pass out in a second. There. So there are 3 ways. One of the things we're doing is we're actually looking at sequential focal planes. We're doing CODEX on one plane, and then we're doing spatial transcriptomics on the adjacent plane. At this point, we're using the 10x Visium, and the resolution is relatively poor, but they claim soon they're going to be getting down to like single cell resolution. Another way, though, is that with the, I believe with the canopy system, you can label oligonucleotides as well. And I think also Akoya has been working with someone to try to incorporate the oligonucleotide labeling of RNA transcripts to combine that with that, so to get in the same image, get protein and RNA transcripts. Connor, do I have that right?
Yeah, I would also add that Akoya is working on adding RNA to their probe lists that would be incorporated. I think that's probably about a year out. So
yeah, so the advantage of the spatial transcriptomics with the 10x Visium is unsupervised. I mean, you to use the techniques that Akoya is putting together, you're going to have target RNAs you're interested in, as opposed to just basically grabbing all the RNAs and finding out what's there. And so there's different strengths and weaknesses. The weakness of the 10x Visium approach is that the spatial resolution is not great. And for that matter, as you get this better spatial resolution, you don't get as much sequence depth, you don't as you do with smaller. But that's the trade-off.
We have one. Oops,
Yeah, go
sorry.
ahead.
We have one other question in the chat that's shifting this just a little bit, but John Phillips is asking, how hard is the process to make the antibody conjugates?
I'll get to that.
Perfect. Thank you.
Ken, I also have a question, and you might also get to that. But what's the throughput, or how fast? Like how
Okay.
long does this washing
I'll
take?
get to that too. Yeah, no, that's not something you talk about quickly, unfortunately. All right. Yeah, I'll get there, I promise. All right, this is an example of something that was just recently done in pancreas, and I'm going to go through the sequences: HLA-ABC, glucagon, HLA-DQ, CD11C, CD3, insulin, and now the nuclei, which kind of obscure everything, but it shows you where all the cells are. And so we've got various people here at IU that are using this to try to understand inflammation and diabetes as well. So with respect to the bone marrow, Eddie Sroor and Kaneli Sarnak, who's on the call, by the way, and Connor Gilbronson have been working on this for a while, and they've developed an antibody panel. And essentially what they're doing right now is doing kind of a template study to develop a CODEX imaging the bone marrow. They're addressing the question of whether or not the relationship between hematopoietic stem cells, osteomax, osteoblasts, and megakaryocytes changes as a function of age. And in order to identify these different cells, you require the ability to identify them on the basis of presence or absence of 13 markers. And that's what this antibody panel is over here. So what they're using an approach of using CODEX imaging of formaldehyde fixed, paraffin embedded 5-micron cryosection tibia from mice of different ages. And we've just really started doing this. And so we don't have a lot of data yet, but we're still in the process of developing the technique, and it's taken a lot of work. We've had problems with bone autofluorescence that had to be dealt with, problems with fixation, problems with the sectioning, and all these things that require serious optimization.
Ken, there is a question in the chat from Mark Buffetto. Mark, do you want to ask it?
It's probably better if you just say it. I'm having weird audio issues.
Okay, I'm happy to read it. It says, what do you use as what they would call a negative control for CODEX?
A negative control. Well, I can talk about how we validate the CODEX, and I'll get to that later too. I hope that covers what you're talking about.
If not, we'll ask it again.
Yeah, I'm sure you will. Good. Perfect. Anyway, so this is an example of what the images look like when you have all the nuclei in place, and this is what happens when you get rid of them. So there's CD45, CD41. These are actually validation studies. So the first thing that Karnak and that Sonali and Connor did was analyze spleen to make sure their antibodies were working properly. And this is CODEX analysis of all these different probes in the spleen. This is the composite of 7 probes. They did 13, but I can't show 13 different probes in spleen. Having satisfied themselves that the antibodies are working in the CODEX, the oligonucleotide conjugates are working properly, then went forward and started working in bone. This is an example of the DAPI image collected from bone. On the left, you've got the bone marrow space, and on the right, you've got the bone. And then they did the same analysis. Again, these are the 6 different markers, another 6 markers, final marker, and a combination of 7 different probes. And based on these studies, Sonali started to try to identify the cells. So one of the megakaryocytes would be classified as being CD45 positive and CD41 positive. We have software. This Halo software allows you to gate the cells, much as you would with a gating strategy in FlowJo or something, to identify specific populations of cells. So these are multinucleate cells that are positive for both CD45 and CD41, and you can see them on the right, shown here. And these are osteomacs, which are identified as CD45 positive, F480 positive, and they're near the endosteum. This is much more complicated. These are hematopoietic stem cells, and they're going to be Lin negative, which is to say they're going to lack all these different probes. They're going to lack, this was clearer when I saw it before, but they're CD117 and CD150 positive. So they're positive for these, they're negative for these. And this is the cell in this particular high magnification field that they found, that would be hematopoietic stem cell. And then osteoblast, which again are Lin negative and SCA1 negative and CD166 positive. So lacking these probes, having this probe, and these are the cells that they identified in the endosteum. So reviewing this, this is the example of it's the summary of the imaging. These are megakaryocytes, these are osteomacs, these are hematic stem cells, and these are osteoblasts. And they did a neighborhood analysis using the Halo software to identify the distances between all pairs of cells identified in the field, and demonstrated that in this particular sample, the distance between hematopoietic stem cells and megakaryocytes was larger than the distance between hematopoietic stem cells and osteomacs. These are then serve as baseline measurements obtained from a young mouse if they're going to test the hypothesis that these distances increase with age. And if you have a question about the science behind that, you can ask Sonali about that maybe at the end of my talk. So getting at the real details of how this all works and the things you have to think about when you start doing this work. So the first thing is that any of these iterative techniques that require lots of rinses, they limit the analysis to 2 dimensions. And the reason for that is that there's no way that you're going to get through all these different rinses if you've got a 3D tissue. So the tissues are 5 microns thick. And if you were to go thicker than that, you would extend the period of time that you have to do your rinses of all your solution changes. So the first question you have to ask yourself is, do you need 3D data? The second thing is that since you have to do sectioning, you're going to have to preserve your tissue in a specific way. And does the tissue fixation or the sectioning alter the tissue morphology? And there are people who tell you that it will in the bone marrow space. The bone marrow space is like goo. It's not especially well preserved. So it's another thing you have to ask yourself. And then the next issue, and that's something that Jörg brought up, is how long does each cycle take? How long will it take to collect each? You're talking about a centimeter scale image that you're going to image iteratively, 3 at a time, and you look at 30 or 40 probes. It takes a very long time. So the question is, will your core, if you've got access to this kind of technology, do you have access to a microscope to give you that kind of time? And I mean, as I'm going to show you later, right now, the CODEX system we're doing, we've got samples that run for 3 days. And that means the system is occupied for 3 days. And no one else can use the system. And so.
Hey, Ken.
Yeah.
This is Diane. Just a question for investigators who are not at Indiana. Is this CODEX imaging, like, would an individual be able to come to Indiana to sort of learn this methodology? I'm just trying to envision how people outside of Indiana would do this.
I think the plan, and this is very much a work in progress, is, and I'll get to that too, but the idea, I believe, is that people would send tissues to us and we would do the analysis here. That said, at the end of the day, it's something that, depending on the kinds of results they get here, I would love to see people incorporate that, and go out and get their own phenocycler systems. Anyway, with regard to speed, we bought, I think, what I would call, we beta tested essentially. We bought a first version of the CODEX system, and there was lots of crude shortcuts on the system that was, in retrospect, were problematic. Akoya has just now recently introduced this PhenoCycler system. It has some significant design improvements. For one thing, they didn't have a microscope originally, and now they've got an integrated robotics and imaging system. And the Akoya system, at the end of the day, really is just a robotic solution changing system. That's all it's doing. And then it's integrated with a microscope system. So the original system, the way that it would work, it would collect a whole 3D volume, and then after you've done all the image collection, it would go through all those different images and identify what it thought was the best focal plane. And then it would fill the mosaic from those things. It's now integrated that into the collection. So it's now collecting one focal plane rather than a stack. It's also collecting from a larger field of view. So it's 2 times larger field of view. Now, the advantages of that is it's like 5 to 6 times faster. Because you're not collecting a stack, you don't have all this image analysis time to identify the proper focal plane. And also because you're doing 2 times larger field of view, you can get twice as many samples going through. So previously, studies that would take 3 days will be completed in something under a day. Now, in addition to being more throughput, that also means you get faster feedback for optimization. Because if you think about it, if you're looking at 50 different antibodies, if you've done immunofluorescence, you know that you have to fool around with antibody concentrations. You have to fool around with integration times. When you do one of these runs, it has settings for how you're doing it. And you may discover partway through that you just simply didn't integrate the image long enough. And so there is a good degree of optimization that has to happen. So it's really crucial that we be able to get these studies done faster to get up to kind of home in on the proper setting. The other thing is obviously by collecting a single focal plane instead of a stack, you have a lot less data, and it ends up being less costly. And I'll get back to that in a minute. And then the other thing you have to ask yourself for all these techniques, and again, I'm kind of embracing all the different techniques that people use. So is this canopy system, is this business of stripping antibodies off, bleaching chemically through the IBEX system? Does it work? The whole idea of this is that it only works if first of all, you can strip off the probes when you're done imaging them. And it only works if after you've stripped off the probes, the epitopes are still available and accessible and have the same avidity for your antibodies that they did at the beginning. So the 2 issues that we got to think about is epitope stability. So for example, in the technique where you strip off the antibodies with these sequential glycine urea, over time, the tissue gets tuckered out and the antibodies don't recognize anything anymore. The same kind of thing with the bleaching reagents is that things kind of change. Cycle carryover is how effectively can you actually bleach or strip your reporters. With respect to CODEX, and this isn't a real advantage of the CODEX technique, is that it seems to be completely cycle independent. In other words, here's 3 antibodies that have been added to a tissue at cycle 4, or they did it at cycle 10, or they did it at cycle 20. And not only did the image look similar, but they did a line profile, and you can see the intensity distributions are essentially the same at all these different time points. And the number of cells they identified, depending on it was independent of the cycle at which they looked for them. So it looks like it's cycle independent. And this is a real nice demonstration of that.
I think that addresses, sorry, I'm going to just interrupt there. Carolyn had a question about how well the focal plane automation is working for you on bones. Was that image before bone so that you could say that the focal plane automation is doing well?
We don't have any idea how the focal plane automation works yet, because it's something that right now we're believing Akoya ad copy. So I can tell you that the current system uses software to identify the appropriate focal plane from a stack of images. And that works reasonably well. It's gotten better over the last 2 years. The new system is going to basically identify that automatically at the capture step. And so we don't know. And we'll be finding out probably in something on the order of a month when we get the PhenoCycler system. And everything kind of depends on that. That's a really good question. The other thing is, are you degrading the tissue such that you're losing cells over time? And this is something that came out of this Nature Protocols article, which I really would suggest people look at, where in cycle 1 and cycle 26, you have exactly the same number of cells of each, all kinds of different tissues and all kinds of different cells. So it seems as if the process of labeling, the sequential labeling and stripping of tissues for the CODEX doesn't change the tissue over time.
And
We-
another question is this run at room temperature? Is there cooling? Is there?
Yes, I'm laughing actually because it turns out cooling is a real problem. So we had a room. So what is the temperature that DMSO freezes at, Connor?
It freezes at 66 degrees Fahrenheit.
66 degrees. So you don't want to let your room get to 66 degrees because everything just goes to shit. You know, none of the solutions flow, none of that stuff happens. So we thought 66 degrees is good for microscopes. They like that, right? You like a cool room. You don't like a cool room for CODEX. So we had to actually go after them. And now Connor's living in tropical conditions in the CODEX system in order to keep our DMSO liquid. You learn these things. Coy didn't tell us this. So anyway, yeah, this is all done at room temperature or better, I guess. So with respect to carryover, this is something that everybody has to evaluate in their own tissues. So there's a question of autofluorescence and there's a question of carryover. So this is a study again done by the Nolan lab, where they did 16 cycle CODEX study with 2 antibody reporter pairs, CD21 and CD3. Kind of hard to understand, but basically this first cycle, they had nothing but the CD3 antibody, and they did not have CD3, CD21, but not CD3. In the second round, they stripped off CD21 and added CD3. Third round, they stripped off CD3, added it back, and added back CD21. And what you can see is the signal goes up and down with each cycle. The signal is constant across cycles. And then they had it 2 sequential cycles for the CD3, and again between cycles it's gone. And one thing I wanted to tell you about this is that it works really well, but even to the degree you end up with individual punctate objects, they're really easy to find. I mean, in the data itself, what you see is these aberrant objects that have combinations of probes that are impossible. So if you design your assay such that the sequential step is going to involve using the same channel for 2 probes that don't have the co-distribution, you can pull these out in the analysis very nicely. So there's a small fraction of carryover, but it's really easy to deal with even in the data.
And so do you guys as a core advise investigators on what sort of combinations?
Yeah, and again, I will get to that too.
Sorry.
It's all right. All right, so this is an example of to give you a sense of what carryover looks like. This is CD31 labeling, and this is CD45 RO. And there are a few structures, a few objects you can see in the field that are constant as I go back and forth between the two. But by and large, the bright stuff here goes away and it's replaced by a completely different distribution. To the degree there is a subset of objects in here that show up, as I said, they'll show up in the scatter plots, they'll show up in T-SNE plots, and they're very easy to pull out. I mean, obviously you can't have too much carryover and deal with it, but it's to the degree it's a modest number of objects, it's very easy to deal with. This is the corresponding CD11C, and you can see a completely different distribution. And these are, what I'm showing you is sequential steps of the same probe. I'm sorry, I should have to explain that. This is like cycle 2, green channel, cycle 3, green channel. This is cycle 2, red channel, cycle 3, red channel. And you can see you're losing the probes. Even the very bright things are completely disappearing from the field. This is Neston. Next section of Podocalixin, and then the third, this is a blank. And so we use blanks frequently. There's a blank at the beginning to establish autofluorescence. There's a blank at the end to see if anything is stuck around forever. But adding a blank cycle, it takes up more time, obviously, but it's a really good thing to do to identify carryover. And the next question is if you're dealing with 20, 30, 40, 50 probes in tissues that are across millimeters, many millimeters, and you're imaging them at like fractions of a submicron resolution, it's an enormous amount of data. It's an enormous amount of data to deal with just as bits, but it's also an enormous amount of data you're going to have software that can analyze huge multiplex images, and you're going to end up getting multi-parameter data that requires special techniques for analyzing it. And with respect to this, we've got 4 software solutions that we're using. One of them is some software that we developed ourselves that is kind of a soup to nuts and analysis. So it's an ImageJ plugin. It supports visualization, processing, segmentation, quantification, supervised and unsupervised classification. And it measures a variety of cellular metrics, and it does neighborhood analysis. And the nice thing about it is that it is all in one platform. So in other words, if I'm looking, if I identify I do an analysis and it turns out that my data look like crap, I can go back and change my processing and my cell segmentation parameters to refine them, if you will. To the degree I get a set of cells that are interesting, and these are not necessarily interesting, but in a scatter plot, if I identify a set of cells, I can put a gate around that, and it'll show me an image where these cells live. If I'm interested in interrogating a particular region that looks interesting for some reason, I can see the data in that region. It's all integrated into one workflow. So it's very nice. And you can do the same thing with the T-SNE plots as well. And this is an example of an analysis of a 36. That was not CODEX data, but this was a VT analysis of a 36-plex CODEX image from human kidney biopsy that supported an unsupervised classification of 9 different cell types in T-SNE plots. So VT is available online. It's an ImageJ plugin. It's constantly being developed. It's a nice tool. It's a nice tool, but it's one of these things that I don't know how many of you have interacted with science, with lab-based software. It's something that there are shortcomings with respect to the interface that a commercial product might not have, if you will, but it's a very powerful technique. Another set of software comes from the Nolan Lab. And I'm not familiar with the software other than the fact that I can tell you that it's available and it does a lot of the same kinds of analyses. And in fact, if you look at the Nolan papers, and at the end of my talk, I'm going to show you all the different Nolan papers you should go read. They use, I think, exclusively this software, which again is available from GitHub. And then finally, we've got software at the CCEH. The Akoya MAV software is no longer being supported because Akoya moved on, but I believe we still have the software. The software that we use most of the time, though, aside from VT, is this Halo software, which is very, very powerful, very expensive, but it's something that you can use through the CCEH working with us. And then this is complicated, and I hope I have enough time and energy to get through it. But the cost-reward ratio has to make sense. First of all, if you're gonna characterize tissue with respect to 20 or 40 or 50 antibodies, it's going to cost you a fortune just to buy the antibodies. But the other thing is that, especially with the Akoya CODEX system involves these costly reagents. I like to refer to the CODEX system as like the inkjet printer model, where the system itself costs a certain amount, but the reagents cost a fortune. And so it's something you have to look at the cost reward benefit. And I think that this is why the CCEH core makes a lot of sense. And I'll explain that to you in a minute. So to just take your breath away for a moment here, 24 probes. Listen, they sell conjugation kits. If you conjugate the antibodies, maybe they cost $700 each by the time you've got the conjugates. You buy the antibodies, you make the conjugates. So you're talking about $16,000, $17,000 in probes. There's a certain amount of reagent costs involved in these labeling the tissues, in the stripping and the reprobing. There's computer time that you have to pay for. So at the end of the day, each run costs $2,097. Total cost for one sample is $19,000. Now, your costs are one-time costs. I mean, a lot of the cost is that antibodies. So to the degree you start doing replicates, you do 3 replicates, it's not 3 times the cost, obviously, because you already bought the antibodies. So this is pretty breathtaking. And so again, one of the first things, the costs don't scale with probe number. So there's a limit, there's a certain cost per run. So it doesn't scale with probe number. It obviously doesn't scale with replicates because you've got this one-time antibody cost. So for example, if you do 3 replicates of 2 conditions, then it's costing you $30,000, not 6 times what one cycle would cost. And the other thing that's important about this with respect to the new system we've got is these costs all assume 1 sample per run. Now, the new system has twice the area. So what that means is that you effectively have the cost of the run. So for 2 samples, you get twice the area. So you're going to actually spend $18,000. Most of your cost is from the antibodies. But 3 replicates of 2 conditions now is down by $7,000. But I think, and the reason that I bring up the CCEH as being an important part of this is that what we're gonna do is we're generating a set. I mean, Sonali and Connor have validated 13 antibodies. So there are more that they're currently validating, and these are going to be available to CCEH members. So all of this cost here. So there will be basically a panel of antibodies you'll be able to use that will run for you. And then to the degree you've got some special protein that you're particularly interested in, you may have to make some of your own antibodies. But at the end of the day, we'll provide a lot of these things. So a lot of this cost will get offset by that. And we plan to grow that up and have more and more antibodies available to people. So, and this is not a sustainable model in the sense that we can't support research project this way. But what we can do is we can, you put together one of these type A or type B funding applications and get funding for that. You should be able to get enough preliminary data to write this into a grant and then make it sustainable, and ideally even get a CODEX system for your university. So at the end of the day, I think CODEX is a very expensive thing for an individual lab to get into unless they really know that's what they want to do. It's an expensive thing to do in a core unless you're getting some kind of support with respect to the cost of reagents and such. So how do you do it? Well, the first thing you do is you decide on a tissue preparation. You're going to do fresh, frozen, or paraffin embedded. And you have to decide that up front because it determines which antibodies you're going to use. You can obtain your antibodies from us. You can get them directly from Akoya, and there's an increasing panel of antibodies for human or mouse, FFPE or fresh frozen available from Akoya, or you can buy antibodies from other vendors. First of all, Akoya has provided a list of antibodies that they've found people have had good success with. There's a website and there's a group in Vanderbilt who also provides a list of antibodies they've worked with. But if you go out and buy the antibodies, the thing we've determined is that they need to be carrier-free. And that's the most crucial thing for successful conjugation. And then you purchase the conjugation kit from Akoya, which includes the corresponding reporter. Someone asked earlier how hard to do the conjugation. The conjugation is not hard. When we first started doing this stuff, I saw a seminar by Yuri Goltsev, who's one of the people in the Nolan lab, and he said that the success rate was like 50%, which was hideous. I mean, the idea of spending $700 and having a 50-50 chance that it's going to work was not good. We've had way better luck than that. I mean, we rarely see a failure. The conjugation is not especially difficult, and then you get through what you have to do. So after you've designed, and when in designing your panel, you try to fill all 3 detector channels. So in other words, don't go out and get a corresponding reporter probe and they're all green. You want them to be green, red, and far red. If you section your tissue and get a reference tissue you can use to as positive controls for your different things, you section them to 5 degrees, 5 microns. Then you validate your antibodies on your tissue and your reference tissue. For conjugates you prepare yourself, you verify conjugation with gel electrophoresis. And then you compare CODEX to indirect immunofluorescence with the unconjugated antibody. You take half your antibody and just do indirect immunofluorescence, compare what you get with CODEX. For all the conjugates, you should compare the CODEX to indirect immunofluorescence with the conjugated. So in other words, you've got this oligonucleotide conjugated antibody. You should do this conventional immunofluorescence with that and compare it with CODEX. And this is the kinds of things that we've done here with the antibodies we've validated so far. You're not done though. You then conduct pilot studies in which you optimize the settings. So that's antibody concentration, the imaging integration time. So you fill the linear range detector without saturating, and checking for cycle dependence and carryover. Once you've got all that lined up, you're all set to go. It's a lot, but in the end, the data you get out of it's a lot too.
One of the questions that's in the chat was for validating, is that validation, do you do any kind of other validation like you do flow or something like that?
So if you look at this paper, this black paper, she describes using flow cytometry as well. It's not something I'm as familiar with, so you certainly can. And you can validate your results with flow cytometry as well. These are easily the best papers. And I think the first paper was by Goltsev. He actually used a slightly different technique, believe it or not. Schurz was the first example of actually using the technique that's available from Akoya. This describes the technique really thoroughly in terms of a real protocol. And these are more like valid. And this is a really nice review article about just general techniques for doing multiplex analysis, and includes stuff about the analysis. things you have to think about for any multiplex study. So that I recommend all these papers really strongly. So the point is, we have the resources here. The core will provide this as a surface. I don't know at this point how sustainable this is, because I have no idea if anybody's going to want to do it. I mean, as of now, we've got 9 people showing up on our seminar. That's encouraging. Someday, there may be more people that we can deal with. So right now, the idea is we're going to support as many studies as we can support. And to the degree that we have to get more systems, and again, we are buying a second system because there's so many people that value doing it. But the other thing we can do is we can support people who put together applications for their own system. So there was a time that I was not convinced of the value of the CODEX system. Akoya was faster to market than their science warranted for a while. But at this point, I think it works really well. There are ways to make it less expensive. There are ways to make it faster. And I think that it's a very powerful technique that could be really valuable to the CCEH, and I hope people start coming to use it. Yeah, that's it. So does anybody have any other? Did I cover all the questions you guys wanted to have answered?
I think so, but I'm going to just invite everybody to show themselves and unmute themselves and pop in for more questions. One of the things I was thinking as you're giving the talk, Ken, is this seems like a great tool to actually look at fetal liver in the mouse and then bone marrow and look at the, you know, the cellular context of what's regulating hematopoiesis. And that seems like a tool that we don't have currently. So it seems very applicable. Mark. Anybody else have questions, comments?
And one thing I want to point out is that the bone marrow is easily the hardest challenge that we've had to address. We've done a variety of different tissues and had much fewer problems. I mean, it's the question of sectioning bone. And it's the question of maintaining the spatial organization of the bone marrow, despite the fact that it's jelly. As opposed to something like the liver, for example, or the kidney. We haven't done liver, but that said, I think that these other organs would be a good deal less challenging.
Cool. Any other questions?
Yeah, I have a quick question, Ken. So the Akoya system is you actually have the slice of tissues on a glass plate, the glass plate, correct?
It's a specific slide that fits into the Akoya holder. Yeah, but it's on a glass cover slip.
So how big? What's the limit of the size of the tissue section?